Hello everyone, welcome to another edition of Hematology and Blood Transfusion Science Lecture Series. This is a YouTube channel where we discuss different topics in hematology and blood transfusion science. Thank you for being part of what we're doing so far. Thank you for your feedback. Thank you for your questions. Thank you for subscribing to our channel. And if you have not yet subscribed, this is the right time to do it. Please hit the button now. Today we will be discussing another important topic, haemopoiesis. My name is Ifeyiwa Mary Ann Okafor, an associate professor of hematology in the Department of Hematology and Blood Transfusion Science, College of Medical Sciences, University of Calabar. It is expected that at the end of this lecture, you should be able to define haemopoiesis, mention hematopoietic sites, define hematopoietic growth factors, and also explain its functions, and understand and explain stages in hematopoietic cell development. What is haemopoiesis? Haemopoiesis is simply the formation of blood cells. Haemopoiesis is simply the process by which blood cells are formed. Remember, formed elements of blood are the red cells, which can also be called erythrocytes, the white cells, which can also be called leukocytes, the platelets, which can also be called the thrombocytes. So, the process by which all these blood cells are formed is called what? Haemopoiesis. It's a general term that is being used to describe the process by which blood cells are formed. In some textbooks, you will see it as haematopoiesis, while on others, you can have them written as haemopoiesis. Sites of haemopoiesis. Adult hemopoietic system originates in embryonic sac. In adults, we know that blood cells are produced by the bone marrow. But remember, we all come from embryo. So, at embryonic stage, the site of blood cell production is the yolk sac. And then we have, as the embryo grows, there is sequential migration from the yolk sac to the liver, to the spleen, and then to the bone marrow as the embryo develops. So, let us look at the blood cell formation before birth. Blood cell formation before birth, what we call the mesoblastic stage, occurs in the yolk sac and mesothelial layers of the placenta. And this occurs at about 0 to 2 months of gestation. This occurs up to about 2 months of gestation. And then gradually, you have the liver and the spleen taking up the work of blood cell production from these 2 months of gestation to about 7 months. And this stage, we call it hepatic stage. So from the mesoblastic stage, which occurs at the level of embryonic stage, to hepatic stage, at the fetal stage of development, you have the liver and the spleen taking charge of blood cell production. And then from about 5th month upwards, you have the bone marrow being involved in blood cell production, and the liver and the spleen slowly relinquishing this function over to the bone marrow. So you see that the baby is born with the bone marrow being the predominant site of the blood cell production. So another stage is what we call the myeloid stage. The myeloid stage is the stage where the bone marrow is chiefly involved in the blood cell production. So you have from the third month onwards, the bone marrow gradually becomes the principal source of the red blood cell and the site for red blood cell production and the others, while the liver and the spleen slowly relinquishes this function to the bone marrow until the bone marrow completely takes over the charge of production the blood cells from 7 months upwards, and the baby is born predominantly with the bone marrow being the site of blood cell production. So exclusively at the point of birth, the bone marrow is the site of blood cell production. The bone marrow is the sole site of effective haematopoiesis in adult human beings. The bone marrow is the sole site of effective haematopoiesis in adult human beings. When the bone marrow is compromised in an adult, you see what we call extramedullary haemopoiesis. This other side, like liver and spleen, can resume back that function that they did at the fetal stage of development. But in a normal adult, the site of blood cell production is the bone marrow. In a normal adult, its production amounts to about 2.5 billion red blood cells, 2.5 billion platelets, and 1.0 billion granulocytes per kilogram of body weight. Imagine this turnover, that the body has to turn over these cells as much as possible, because if you remember, the lifespan of these cells, like red blood cell, takes about 120 days. For platelets, it's about 7 to 10 days, while granulocyte is just about a few hours within the circulation. So there is need for the system to have a high rate of turnover, production of these cells, in order to replace those that have been catabolized and taken out of the system. Blood cell formation at birth. At the bone marrow, when a child is below 5 years, all the sites, all the bone in the system are involved in the blood cell production. All the bone in the body is actually involved in blood cell production. By the age of 18 years, hematopoietic marrow is found only in the vertebrae, the ribs, the skull, the pelvis, the proximal epiphysis of the femora, and the humeri. So by the time the child is growing, at the end of adolescent stage, we find out that the marrow, the bones that are involved in blood cell production, are restricted only to the vertebrae, the ribs, the skull, the pelvis, and the proximal epiphysis of the femora and the humeri. Let us look at the summary of the sites of haemopoiesis that we just discussed. We said we have 3 stages. The mesoblastic stage, where you have the yolk sac and mesenteric layer of the placenta being the site of blood cell production. And then we have the hepatic stage, where you have the spleen and the liver being the site of blood cell production. And then you have the myeloid stage, where you have the bone marrow being the site of the blood cell production. And then in fetus, you have from 0 to 2 months, you have the yolk sac being the site of production. From 2 to 7 months, you have the liver and the spleen being the site of production. And then from 5 to 9 months, you have the bone marrow being the site of production of blood cells. While in infants, you have all the bones, the bone marrow of all the bones being the site of the production. While in adults, you have only the vertebrae, the ribs, the skull, the sacrum and pelvis, the proximal end of the femur and the humeral, the sternum and the ilium being the site of the production of blood cells. The blood cell production is restricted to the marrow of these bones, because as the child is growing from infancy to adolescence and then to adulthood, the red marrow is being replaced by the yellow marrow, and the yellow marrow is not involved in blood cell production. It's only the red marrow, which is found in the cells, the bones I have just mentioned. Let us look at haemopoietic growth factors. What are haemopoietic growth factors? For bone marrow to actually produce these blood cells, the bone marrow is also structured in a way that it has all the necessary ingredients and the things needed for it to actually produce these blood cells. Among other things that the bone marrow contains is what we call growth factors, haemopoietic growth factors. And these haemopoietic growth factors are naturally occurring hormone-like glycoproteins. You can say they are cytokines that are required for initiation and sustenance of normal proliferation and differentiation of haemopoietic progenitor cells and the formation of mature blood cells. These haemopoietic growth factors are needed, and they are natural occurring hormone-like glycoproteins. You can call them cytokines that are required for initiation, sustenance of normal proliferation, differentiation of haemopoietic progenitor cells, and normal function of mature blood cells. These haemopoietic growth factors act at different stages, different parts of bone marrow to actually bring about their function. There are ones that act on the stromal cells. You have the interleukin-1 tumor necrotic factor. We have some that act on the pluripotential stem cells. We have the stem cell factor, Fick ligand. We have the ones that act on multipotential progenitor cells. Example is interleukin-2, granulocyte megakaryocyte colony stimulating factor, what we call GM-CSF. CSF is colony stimulating factor. And we have interleukin-6, we have granulocyte colony stimulating factor, and then thrombopoietin. We have others that act on committed progenitor cells. The others that act on committed progenitor cells, and we have examples of these ones are the granulocyte colony stimulating factor, the megakaryocyte colony stimulating factor, interleukin-5, which is a eosinophil colony stimulating factor, and we have erythropoietin and thrombopoietin. What are the functions of these growth factors? Number one, they are involved in the regulation of normal haemopoiesis in the bone marrow. They are involved in the regulation of normal haemopoiesis in the bone marrow. And they are also involved in the induction of cell viability and cell multiplication. They are involved in prevention of apoptosis, that is programmed cell death. Apoptosis is programmed cell death. So they are involved in the prevention of apoptosis of haematopoietic progenitor cells. Haemopoietic progenitor cells, they prevent apoptosis of haemopoietic progenitor cells. And they are also involved in stimulation of phagocytosis of bacteria. They are involved in the stimulation of phagocytosis of bacteria and also enhancement of tumoroidal activities of macrophages. Enhancement of tumoroidal activities of macrophages. Now, the haemopoietic system is a hierarchy of cells in which multipotent haemopoietic stem cells give rise to lineage committed progenitor cells, which divide to generate the maturing and mature blood cells. It is good for you to note that the hemopoietic system is a hierarchy of cells in which the multipotent hemopoietic stem cell give rise to lineage committed progenitor cells, which divide to generate the maturing and also the mature blood cells. Morphologically, hemopoietic stem cells are undifferentiated and resemble small lymphocytes. They are undifferentiated and they resemble small lymphocytes. CD34 is the best known marker of human stem and progenitor cells. CD34 is the best known marker of human stem and progenitor cells. Stem cells are capable of self-renewal. In haemopoiesis, the stem cells, hemopoietic stem cells, which is the main source of every other cell, the unique feature they have is that they are capable of self-renewal and differentiation when they divide and are responsible for producing all blood cells throughout life. They are capable of self-renewal and differentiation when they divide and are capable of producing all blood cells throughout life. When in steady state stem cell divide, only 50% of the daughter cells on average differentiate. The remaining 50% do not differentiate but maintain stem cell number. As hemopoietic cell development proceeds from stem cells to progenitor cells, the probability of renewal decreases and that of differentiation increases. Let us look at stages in hemopoietic cell development. We have the stem cell pool. This stem cell pool, the hemopoietic stem cell. And this stem cell have this unique ability of self-renewal. They have the ability to renew itself. Not only that they renew itself, they also have the ability to differentiate. They have abilities to differentiate into lineage committed cells. Remember, we have the stem cell pool, which can self-renew itself and at the same time can differentiate. When they do, they differentiate it into lineage selection. This lineage selection can either be myeloid lineage or lymphoid lineage. The myeloid lineage gives rise to red cells, the platelets, the neutrophils, the eosinophils, the basophils, while the lymphoid lineage gives rise to the lymphocyte series like the B lymphocyte, the T lymphocyte, the natural killer cells. So we have the stem cell pool being self-renewal and then differentiate into lineage selection, and then this lineage selection now goes on to mature, the maturation stage, and then they exhibit their different functions in the system. The red cell involved in transport of oxygen, that is the main function, generation of energy through transport of oxygen. And you have the white cells that are involved in defense, and then the platelet that chiefly involved in coagulation, blood clot. So you find out that when the stem cell pool differentiate into committed lineage lines, these cells mature, carry out their function in the system, and they undergo programmed cell death when their lifespan is over, what we call apoptosis. And when that is done, they are pulled out of the system, and the process continues. The new cells that are being produced replaces them. Now, through division differentiation, the pluripotent cells mature to multipotent mixed myeloid progenitor cells and lymphoid progenitor cells. Further division and maturation leads to these cells having committed cell lines, and the uncommitted lymphoid cells give rise to the B lymphocyte, the T lymphocyte, and the natural killer cells. And then the human stem cell and most hematopoietic progenitor cells is worthy to note as CD4 positive and CD38 negative. I think there is this particular clip that I want to play now that will help you understand what I am explaining. Please watch the clip.
Blood cells have a limited lifespan and must be continuously replaced through a process called haematopoiesis, which starts in the red bone marrow. All formed elements of the blood derive from a common progenitor, the hematopoietic stem cells, HSCs. HSCs are multipotent, meaning they can differentiate to all types of blood cells. They also have the ability to multiply constantly to maintain their numbers in the bone marrow. Formation of blood cells from hematopoietic stem cells is a multi-step process involving several intermediate progenitors and is regulated by a network of signaling molecules known as cytokines. These cytokines control the proliferation, differentiation, and survival or death of the various progenitors. By doing so, they maintain steady-state levels of blood cells in normal situations, and, in response to certain stimuli, induce production of a particular cell type.
Now, what we have in this diagram, like what is explained in that short clip, the stem cell, which is remember I said this stem cell, hemopoietic stem cell, has the ability to self-renew itself and also do what? Differentiate into pluripotent stem cell, which gives rise to myeloid progenitor cells and then lymphoid progenitor cells. Now, these lymphoid progenitor cells gives rise to erythrocyte colony forming units, which undergoes series of changes to form erythrocyte, which is red blood cell. We also have the myeloid progenitor cell giving rise to what? Megakaryocyte, which undergoes some processes to get to what? Platelets. We have the myeloid progenitor cell giving rise to what? Basophil colony forming unit, and which ends up forming what? Basophils. We have myeloid progenitor cell giving rise to eosinophil colony forming unit, and then we have eosinophil. And then from the same myeloid progenitor cells, we have what? Granulocyte, monocyte, colony forming unit, which give rise to neutrophils, macrophages. Macrophages in the tissue give rise to what? We give rise to monocytes. That is, the granulocyte, monocyte colony forming unit give rise to neutrophils and monocyte. And monocyte in the tissue is what we call what? Macrophages. And then we also have monocytes. We have the granulocyte, monocyte colony forming unit giving rise to what? The dendritic cells, which is an antigen presenting cell. So we have here the lymphoid progenitor cells giving rise to the lymphocyte series. We have in most one, remember, these ones matures in the bone marrow, while the lymphoid progenitor cells, the B lymphocytes in the bone marrow, the T lymphocyte matures in the thymus. In the thymus, the T lymphocyte matures in the thymus. And then we have the natural killer cells also matures in the bone marrow. So the lymphoid progenitor cells give rise to B lymphocyte, the T lymphocyte, and the natural killer cells, while the myeloid progenitor cells give rise to other cells like erythrocytes, platelets, basophils, eosinophils, neutrophils, monocytes. Okay. So this diagram depicts in summary the process of haemopoiesis, the process of blood cell formation, the self-renewing stem cell giving rise to pluripotent stem cell, and then dividing to have myeloid progenitor cells and lymphoid progenitor cells. The lymphoid progenitor cell giving rise to lymphocyte series, the B lymphocyte, T lymphocyte, which matures in the thymus, and then the genotular killer cells. And the myeloid progenitor cells passing through different processes to give what? To give rise to erythrocytes, platelets, which is thrombocytes, basophils, eosinophils, neutrophils, and monocytes, and dendritic cells in extension. Remember that we have the hemopoietic growth factors acting at different stages of this blood cell production to stimulate, sustain, and also maintain these processes. Cellular release. Let's look at cell migration from bone marrow to the general circulation. Cell migration from bone marrow to general circulation, when they are eventually produced in the bone marrow, they migrate to the general circulation. This process is tightly regulated by a complex interplay signals from the bone marrow microenvironment, circulating growth factors, and cytokines, and feedback mechanism that accounts for balanced production of different blood cells. Cellular migration is under complex control of a family of small cytokines termed the chemokines. Releasing factors that are involved in the initiation of bone marrow egress, especially for granulocytes, include granulocyte colony stimulating factor, granulocyte megakaryocyte colony stimulating factor, C3e component of complement, thymocyte activated plasma containing complement fragments, glucocorticoid hormones, androgenic steroid, and angiotensin. These chemokines I just mentioned are all involved in the initiation of bone marrow ingress, especially that of granulocytes. To leave the marrow, the reticulocyte depends on a pressure gradient across the membrane to drive it through the pore. Blood cells pass from the bone marrow into the blood capillaries through the processes called homing and diapedesis. Homing is a process by which hemopoietic cells move to specific tissue and organs in the body. Homing is the process through which hemopoietic cells move to specific tissues or organs in the body. Homing process involves a series of steps, including chemotaxis. Chemotaxis simply means cells follow gradients of signaling molecules like chemokines, like I mentioned, colony stimulating factor, granulocyte colony stimulating factor, and others. Adhesion, ability of the cells to attach to the endothelial cells that line the blood vessels. Extravasation, ability of the cells to squeeze through the gaps between the endothelial cells, and then migration. So homing involves chemotaxis, adhesion, extravasation, and migration. While diapedesis is the ability of cells to squeeze through the pores of the capillary membrane. Diapedesis is the ability of the cells to squeeze through the pores of the capillary membrane. Then when the cells have eventually migrated into the circulation through homing and diapedesis, they now go on to get involved in executing their various functions. The red blood cells that involve in transport of oxygen, the white cells that involve in body defense, the platelet that involve, among other things, in blood clot formation or arresting of bleeding tendencies or coagulation processes. They're involved in those things and they live out their lifespan. And when they leave out their lifespan, they undergo what we call apoptosis. The final stage in the life of a blood cell is death and disposal by what we call the apoptosis, which is programmed cell death. Apoptotic cell death is a mechanism for disposing of unwanted or excess cells, and it occurs widely in biological systems. It ensures the destruction of cells without releasing any lysosomal or granular content that will cause an inflammatory reaction. You should note that the stem cells are the most important cells in haemopoietic cell production. These stem cells are ultimately responsible for regenerating haemopoiesis following damage to the marrow or to the haemopoietic system by chemotherapy or after stem cell transplantation. Remember that the end product of haemopoietic cell development are cells that are highly specialized for their different functions in the body. The end product of haemopoietic cell development are cells that are highly specialized for their different functions in the body. The end product of haemopoiesis is the production of different cells like erythrocytes, that is red cells, platelets, and leukocytes, that is white cell. In our next lecture, we are going to be looking into discussing production of red blood cells in detail, that is erythropoiesis, and also production of white blood cells and production of platelets in detail. I hope you have learned something in today's lecture. Feel free to send in your questions and feedback. And also, don't forget to subscribe to our channel. Join us in our next lecture where we'll be discussing erythropoiesis. Thank you.